The solution to improving acuity and refraction measurement? Introducing the Dyop, the 21st century method of dynamically measuring vision and measuring the effects of color. A diop is a spinning segmented ring used as a visual target. A diop uses vision physiology rather than culturally dependent letters to measure acuity. The smallest diameter diop ring detected as spinning is the acuity endpoint and is much smaller than the smallest letter identifiable in a Snellen test. When using a diop for vision testing, two diop rings are displayed, but only one of the diop rings is spinning. The patient is asked which ring is spinning, the left ring or the right ring. If the patient doesn't detect either diop ring as spinning, then both rings are made larger until a spinning ring is detected. If either diop ring is detected as spinning, then both rings are incrementally made smaller and the spin location or spin direction is randomly switched until spinning by the smaller rings cannot be detected. Diop subacuity is any ring diameter where the spinning ring is too small for spinning to be detected. Increasing the diop diameter from a subacuity size to the next largest diameter where spinning can be detected verifies this larger size as the acuity endpoint. The acuity endpoint is the smallest diameter diop where spinning can be detected. To prevent guessing, false positives are easily detected by asking the patient which diop ring is spinning and which direction is it spinning, clockwise or counterclockwise. Guessing can be further reduced by repeating the test several times, where each time the location of the spinning ring and direction of spin are selected randomly. The result is that diop testing provides a rapid, precise, and accurate measure of acuity regardless of patient literacy, culture, or age. Dyop research has consistently shown that Snellen testing overcorrects acuity measurement by adding excess spherical minus power and may be a contributing factor in the global epidemic of myopia. Inherent with Snellen testing is the problem that we don't see black. Snellen acuity measurement is due to the identification of the white gaps in the Snellen letters and not the black letters. The result is that Snellen and other similar tests have excessively large and inconsistent gaps. The theoretical average Snellen stimulus gap is 1.0 arc-minutes squared. Diop acuity measurement is made possible by the detection of motion of the identical and consistent gaps in the spinning diop. The spinning diop gaps avoid depletion of the photoreceptor response by providing a strobic stimulus. The consistent diop stimulus gap is 0.54 arc-minutes squared. The increased precision, accuracy, and efficiency of a diop is likely due to the size of the diop stimulus gap being half of the area of the Snellen stimulus gap. The significance of that 0.54 arc-minute square size of the diop stimulus gap, otherwise known as the minimum area of resolution, is that it explains the functionality of the neuroganglia as a biological circuit board. There are approximately 200,000 cone-shaped photoreceptors in the fovea, with a density of 17,500 cones per arc degree. That 0.54 arc-minute square size of the minimum area of resolution calculates to be a diop stimulus area of only 20 cone-shaped photoreceptors. Since each optic nerve fiber combines the response from 100 photoreceptors, then the neuroganglia is functioning as a biological circuit board to combine the stimulus of 5 clusters of 20 cone photoreceptors to stimulate each optic nerve fiber to then create the specific color and intensity of the signal sent to the brain. As the color pattern of each cluster of 20 cone photoreceptors crosses over the fovea, it also provides an indication of motion across the visual field. That motion detection ensures that vision is dynamic and autonomic. As a result of the precision of the Dyop stimulus, Dyop acuity testing is up to 6 times more accurate than standard Snellen testing. Dyop acuity testing is up to 8 times more precise than standard Snellen testing. Dyop testing is typically twice as efficient as Snellen testing. Dyop acuity tests typically take only 10 to 20 seconds per eye. Dyop refractions typically take only 90 to 180 seconds per eye. Dyop vision testing is also possible with infants as young as 14 weeks of age. Large groups may easily and rapidly use Dyop acuity testing by comparing the Dyop diameter where spinning is first detected versus the Dyop viewing distance. Dyop testing for low vision is inherently more precise than Snellen testing because the Dyop test doesn't have the bloated logarithmic curve of a Snellen test. Our goal is for the Dyop test to become the 21st century standard for global vision testing. Welcome to the Dyop revolution, helping the world see clearly, one person at a time.